Hi Aaron,
Hi Bronwyn.
welcome to my room.
It's a pretty fancy lounge room.
It is, and it's even better than that because it's a wandering room.
What's a wandering room?
Have a look out the window. Look,
Where are we?
where do you think we are?
That looks like Antarctica.
That might be right.
This is doing my head in.
Hi guys,
Hi, I'm Aaron.
and I'm Bronwyn, and we're here today to talk about week 7's prac.
Alpha amylase and the breakdown of
Starch and
starches?
glycogen.
Yeah.
Alpha amylase is found in our mouths.
Yep, it's a enzyme in saliva which cleaves the 1,4-glycosidic bond.
Yep,
From
it does
memory?
indeed. So alpha 1,4 linkages are found in starch and also in glycogen, and they're used to hold glucose molecules together. So the alpha amylase is breaking down molecules where the glucose is.
Okay, so it's kind of coming in like scissors and cutting along.
Yeah.
Okay.
Unfortunately, it's not very effective, the alpha amylase in our mouths.
Yeah, I've heard that it actually has a very low percentage of breaking those bonds. Then again, your food generally doesn't stay in your mouth long enough for those breakages to kind of occur before
that's
heading for
true.
digestion.
And so once, of course, it goes down your stomach, there are more amylase enzymes down there which will break it down further. But we're looking at the mouth ones today. And everyone gets to test their own.
You get to test your own saliva, or your partner's. Depends on who wants to spit into the tube.
And you'll be surprised because everyone's amylase levels are different, so you'll get different results. So the hypothesis this week, we want to see a lot of variation in what they come up with for this one, don't we?
Well, yes. I mean, just a nice comparison is your amylase activity will be completely different to mine. It is, individual to individual. So
Mine's going to be superior.
most likely, strong, powerful woman.
We're up to glycogen and starch and alpha amylase and all sorts of exciting things. So try and think of something that's more relevant. And of course, last week you guys all looked up. You all looked up references to figure out a really good
level.
hypothesis. So try again this week. See what you can come up with. Astound us.
Surprise me.
It's going to be hard to surprise him.
One that I would probably, and this isn't giving you answers at all, would be something along the lines of the breakage of, starch compared to glycogen. Is there a difference?
Yeah, and of course you're going to expect that because glycogen is how us humans store our glucose. And so it's packed pretty tight. So there's a lot of branches that aren't alpha 1,4. So it's got the alpha 1,4 and 1,6, so both starch and glycogen have them, but there's so much more in glycogen, so...
Will that 1,6 affect how the amylase works?
Probably.
Yeah,
So that's something to think about.
I like it. So there's actually quite a lot to do in today's prac, if I'm correct.
The easiest way to start will be to look at our saliva. So we're going to have to actually spit in a tube. I'm sorry guys, but it's for the good of mankind to spit in that tube and to be able to test your own enzyme. And so once it's in the tube, you're going to be adding either starch or glycogen into the tube. So you'll have 2 tubes, one with starch in and one with glycogen in. And then you get to do a time course.
Now, the time course is where you're actually going to be monitoring the amount of starch or glycogen broken down within the tube over a set period of time. So you will obviously have your reaction running and you'll actually take out a aliquot every 3 minutes. So make sure you keep your timer running and every 3 minutes you're going to take out an aliquot.
And the second step is to figure out roughly how much glucose is actually in the starch sample and the glycogen sample that we're using the alpha amylase to break down. So that's a second set of experiments, and again they are paired. You have to do it once with the starch and once with the glycogen. And the lab staff have actually pre-prepared that, so it's called a hydrolysate of starch and a hydrolysate of glycogen. And so those hydrolysates contain a lot of glucose, mainly glucose. And so the whole aim in what you're doing is to figure out just how much of that sample that you have used with your enzyme, if it's broken down thoroughly into glucose, how much is actually present there.
You'll then be required to prepare a series of glucose standards for analysing the amount of glucose from your starch and glycogen samples.
So that includes the ones that you did the time course on, so the ones that are based on your own salivary amylase, as well as the total hydrolysate of starch and of glycogen. So you're going to end up with a lot of samples.
Do we know how many tubes?
No, I haven't counted them up. I think that's for them to count up.
There is a lot of tubes.
There are.
Make sure you go through and check everything, because when you come to do the colour development, you're actually going to incubate all your tubes at once, just to save on time.
And just make sure that before you do the colour development, every single tube you have should have 0.8 mils of DNS present in it. And you'll know that straight away because it's orange. And you will then be heating them all for 5 minutes at 100 degrees. And of course that will involve lancets again.
Yes. So see your demonstrator to get a lancet so you can pierce the top of your tubes so nothing explodes and takes out somebody's eye.
So that now that we've outlined the prac, we'll just show you a couple of little videos of what you're going to be doing in the prac. So we're going to start first with this one on the time course assay. Into an empty Eppendorf tube, add 1.8 mils of starch.
Now, you'll need to actually do that in 2 separate pipettings, because obviously we don't have any pipetters which can do that volume in the lab.
And then you'll add your 0.2 mils of diluted saliva. And then of course when you're doing the glycogen assay, you'll do the same thing, but you'll start with 1.8 mils of glycogen instead. As soon as you add the saliva into the solution, start your stopwatch.
At the 3-minute mark, you'll need to remove an aliquot of 0.2 millilitres.
And you'll add that straight into the DNS.
And you can add that straight into your DNS solution.
And then leave the stopwatch running, because you'll also have to do the same thing at time point 6, 9, 12 and 15. After 15, you can stop. So this experiment is quite complicated, as you can see. So once you have your saliva assays up and running, you might want to think about working with your partner so that the partner does something different to what you're doing to actually get it running in time. So I guess one thing you could do is one person do the action of amylase on starch and the other one do it on glycogen, so that then you can work together in making sure that you get all of the assays done on time.
Mm-hmm.
After you have completed the saliva assay, then move on to creating the total hydrolysates for both the starch and glycogen. And that's just a quick bit of pipetting, and it won't take too long to do.
After you've prepared both of your time courses, your hydrolysates and your glucose standards, you'll incubate them for 5 minutes at 100 degrees. This will cause a colour development to be monitored on the spectrophotometer. You'll then need to cool them down to room temperature by placing them on ice. Here are some examples of the colour development that you'll have for reading on the spectrophotometer. There are the time course, the hydrolysates, and the standards.
There's just one more result we're going to show you now, and that's of a spectra showing an example of what you can expect to see when you measure your glucose, which could be from your glycogen or the starch or simply from your glucose standards, of what the absorbance will look like.
At 540?
540 nanometers, yeah. So once you have all of your results, you're going to need to do some work to interpret them. And the first, of course, is to create the standard curve from the glucose standards. So you have 4 standards and a reference, which is also known as your blank. So first of all, you will graph them, and then you can measure and figure out how much glucose is in all of the other samples that you've assayed.
It should be brought to your attention that there is a dilution factor to be taken into account with the hydrolysates.
You'll need to take into consideration how the starch and glycogen solutions were prepared, and that's in a note about the total hydrolysate, and where it talks about the samples were supplied for you and that they were treated beforehand. So they took 0.2 mils of either the starch or the glycogen, added 0.3 mils of hydrochloric acid, and then they added
0.5 of sodium hydroxide.
So you'll
boiled
need
it all up.
Yep. So you'll need to add those up and figure out what proportion of your sample really was the starch or the glycogen.
One last thing, the additional practice questions at the end of your results and your discussion, make sure you're also doing those because they go towards your tick mark.
They also help you think about what you're doing and learn. Practice for the exam.
Practice for the exam.
All important stuff.
Anyway, we'll
So-
see you in the lab.
We will indeed. See you.
Bye.